In the previous video, we did an overview of remediation technologies. On this one, I want to focus a little more on three specific technologies: permeable reactive barriers, cometabolism biodegradation, and surfactant-enhanced remediation. So the example I'm going to do for permeable reactive barriers was for chromium, a metal, and for chlorinated solvents, a TCE. The cometabolism bioremediation was for a specific isomer of a pesticide that just wasn't biodegrading other ways. And then surfactant-enhanced remediation, I'll talk about using it for NAPLs, especially for DNAPLs. So the way permeable reactive barriers work is you have a passive wall here that's not a barrier to flow, it's a barrier to the chemical. So the chemical comes through, something happens in that wall where it gets treated and clean water comes out the other end of it. So this is a little graphic of that. So it's a passive technology. The project I was involved with was with the Elizabeth City, North Carolina. They had a Coast Guard base, and they did chrome plating inside a building there. And then they had leaks of chromium and then TCE, a degreaser that went through. And so they had this plume heading for the river here, and they thought they can put in a permeable reactive barrier, a wall, to stop that. And again, it doesn't stop the flow, it just stops the chemical. So what's going to happen is you put iron filings with sand in that wall, and iron is oxidized, it rusts, and the other things are reduced. The other things are chromium, which in the +6 oxidation state is toxic and soluble in water. If we can get it to reduce by the iron oxidizing, get the chromium to reduce to +3 oxidation state, that's non-toxic, and it's insoluble. It forms insoluble precipitates. TCE, trichloroethylene, is reduced under those conditions through reductive dechlorination, which works actually a little better than in trying to oxidize it. So how it works is you put in iron filings, zero valent iron. There's some mechanisms that happen there. So the iron oxidizes, so it goes up to Fe2+, and in that, the chlorinated solvents, the chlorines come off of that. And that also happens with some of the other oxidation states of iron once it gets started. For TCE, this actually involves, I won't get into the chemistry of it, but TCE is trichloroethylene. So if you go to 2, that's 2-chloroethylene. And then you go to 1-chloroethylene, that's vinyl chloride, which is actually more toxic. And then the chlorine comes off, and then it can go to ethylene or ethene and ethane. So it's several steps to it, and you actually have to get it to go all the way to really clean it up. The incomplete process will produce adverse intermediates. The physical designs of it, there's the continuous PRB, which is just a wall there, or to use less iron filings, maybe we could put in some sheet piling or grout walls and funnel it to one section where it goes through. So one of my jobs was actually I modeled different configurations of it. And it turned out it was just the easiest thing just to put in a straight trench like that. Yeah, they call that the funnel gate system on top, or a single continuous barrier system at the bottom. So I wasn't there when it was installed. I was there before the installation, but this is trencher putting in the trench where they put iron filings in. And I think it's worked. You can look it up. The next case I want to talk about is soil groundwater remediation for an industrial site in the Netherlands. And I saw this in a magazine, and I just thought this was the coolest thing ever. So I contacted the guy, and he sent me these slides. This is years ago. Wow. Well, it won an award in 2001. It was like 20 years ago when I saw it. It's a really cool project. So they thought they had this old pesticide there, and they're trying to get rid of it. Why don't we use the bacteria that are already present and try to figure out a way to get those bacteria to eat the stuff? So it was pesticides. A chemical company made the pesticides, and they were stored there and sat there forever and ever. And then they wanted to redevelop the site. So they reduced the waste. They removed the waste, and then they still had this contamination of this pesticide, lindane, hexachlorocyclohexane. And then that biodegrades, like we saw with the TCE, to monochlorobenzene and to benzene. So they still have the contamination there. So hexachlorocyclohexane, it's not a benzene, it's cyclohexane. Hexa means 6 of them. So there's 6 chlorines stuck on 12 spots on that cyclohexane. And so they can be in different combinations in different places. Well, one of those isomers did not like to biodegrade. The other ones did, but one of those isomers, I don't even know exactly what it looked like, wouldn't biodegrade. So they figured out that this isomer could be broken down under anaerobic conditions if you have another natural substrate as a co-substrate. And again, my analogy is the bugs eat other food and use the stuff as ketchup. Actually, I said that backwards, didn't I? In the other video, the food is what you're giving them. They use the HCH as ketchup. And this company said their substrate is proprietary. They wouldn't tell me what it is. I said, is it like some kind of alcohol or something like that? Something like that. Now, it does break down into benzene and monochlorobenzene, but those can be aerobically degraded. So it anaerobically degrades to monochlorobenzene and benzene, and then you can get it to aerobically degrade to other stuff that's harmless. And this, they did lab tests and they found out this, the HCH, that's what they're trying to get rid of, goes down, the benzene goes up, the monochlorobenzene goes up, but then they're actually able to get that to degrade under aerobic conditions. So they go anaerobic with the substrate that they add, and then they go aerobic and they get rid of it. So what their system looks like is underneath the container terminal here, they infiltrate the electron donor, or that's the goop, the food that they feed it, but they won't tell me what it is. And then they extract the groundwater out of there, and then they treat that aerobically. So you can see this is what it looks like. There's a container terminal built on top of it there. This is a cross section of it. So you can see the layers in there. They have the HCH is kind of up on top there, and then it leaches down through there. They put the extraction drain at the end here, and then I'll show a couple more overhead one here. So the groundwater is moving toward the canal here. They put it in their infiltration. That's where they're squirting in the food stuff. This is built over the top, but they're squirting in the food stuff, and then they take it out of there right before it hits the canal, and then they take it to a wastewater treatment plant on site there where they bioremediate it. So there they are, putting in the system, putting in the pipes that will inject the substrate in there. Another cross section again, water extraction drain there. It's moving toward the canal. And they actually have monitoring wells. Since they have different layers there, you can actually do this with a monitoring well. You can set up for different samplers at different points. So you have tubes going to the top of the well, right? And you know they're labeled which tube is at what depth, so they can sample these different depths and see what the concentrations are at the different depths. And they squirt that stuff in there twice, there and there and there again, as it goes through. And in the field results, it looked like the lab results. They were very happy about that. The HCH went down, monochlorobenzene and benzene went up, but we can treat that in our wastewater treatment plant. It worked. And again, I thought it was super cool. Got information on it. It's kind of an example of the theory of the cometabolism. It actually works. The third one I want to talk about here is surfactant-enhanced remediation. I'll try not to talk too much. This is what our research group did in grad school. And it's for NAPLs. We did a lecture on NAPLs a couple weeks ago at this point, probably. And we have L NAPLs and D NAPLs. But I wanted to show this to show the interface between They napple the oil and the water. The napple is on top for an L-napple, on below for a D-napple. And again, the problem with napples is that they're all over and they're not very soluble, so they're hard to get out. If they didn't dissolve at all, they wouldn't contaminate anything. So it's a problem. And then they go down there and they get trapped in there in little spots. The D-napple goes down to the bottom. You get the dissolved contaminants, and you have the smear of the napples all over the place. Now, a D-napple, you can actually go down and hit a fractured rock and get down in there. So it's really hard to deal with. So surfactants offered a way to do it. So those little blobs are stuck there between the soil grains, and we want to get those out of there. One thing we can do is use surfactants. Surfactants are surface active agents, detergents, and the structure of it is they have an organic, like a hydrocarbon chain, and they have a polar head. So the head likes water, the tail doesn't like water. So because of that, they will accumulate at an interface. They are surface active in the surface between the oil and the water. And they actually do two different things. They increase the solubility in water, and then they lower the surface tension. And different surfactants, different formulations will do this differently. But they'll actually form little tiny, it's microscopic, but little tiny balls in the water called micelles. All the little organic tails stick together, and that creates like little mini droplets of oil in the water, sort of. And so the oil chemical will want to come out of the napple and get in those little balls. So I did these tests. These are tests I did years ago where I have PCE, tetrachloroethylene, stained red in these columns. I'm going to run water through it and then two different surfactants. And you can see the surfactants are different. It's dissolving out. So after 8 pore volumes, so I take the pore volume of this column and 8 times that of the surfactant solution is getting this one about a quarter clean, starting to get this one, and the water isn't actually clean at all. This is for 35. At that point, this surfactant, it's about half clean. This one's pretty much all clean, and the water doesn't look any different than it did at the start. So you can see by increasing the solubility, we can get the chemical out of there a lot faster. The other thing that surfactants do is they stick their little tails in the droplets of the oil, and that reduces the interfacial tension, and they're stuck in between there by capillary forces, but they stick their little tails in there, and then those blobs will come out of there. So I did the same thing in experiments with surfactants formulated for mobilization, and I can get the stuff out of there in just like 1 pore volume or something. So this is actually technology that was used in enhanced oil recovery. Well, why not do that? Well, you got to control it then. That's the problem. It might go down deeper. For a field system, you pump the surfactant in, then you got to pump it out. It's real tricky. I'll say easy, huh? It's real tricky with get the chemistry exactly right. You got to be able to contain your surfactant and your contaminated water. And for the economics of it, you got to recover the surfactant, reuse it if you can. And then you got to figure out what to do with the water that's coming out. So it's a pretty tricky system, actually. And the economics of it and optimization of it depends a lot about the above ground processes and recovering the surfactants. I'll show you a system in a second here. And the regulatory permitting, you got to get permission to put the surfactant down there, and then you got to get permission to do whatever you're going to do within the treated water. So this is an example of a system, and these are done. Now my professors actually started a company doing this stuff, and you put the surfactant in, and then you get the stuff out, you separate the oil and the water, and then now you want to try to get the surfactant out of the water, and you got to treat the contaminated water. So it's a pretty tricky system, but it's really cool. Like I said, I was deep in that for several years. All right, summary. Surfactants are good for napples, especially for D-napples. Why D-napples? Because it's more expensive. Surfactant-enhanced remediation is expensive. For L-Napples, there's other stuff like this SVE air sparging stuff to get it off the top of the groundwater. The D-Napples are down there. SVE is not going to work for that. Cheaper methods are available for other things. The D-Napples, it might be worth the complication expense of it. It is chemistry intensive. You got to do your homework on the site chemistry and on the chemistry of surfactant, on the chemical stuff. And it does involve above ground processes to make it all work. And to jump back to a summary of the permeable reactive barriers, it can be good for certain metals and organics. The attraction of it is it's passive, no extraction. You don't have to get permission to do anything with the water, re-inject it or anything. It's limited by depth. I mean, you can only go as deep as you can put in a trench. Biodegradation in general, any organic will biodegrade. I put a question mark there, but really, some are really, really hard to do. But that company in the Netherlands, they got creative and they found a way to biodegrade something that had been there for 50 some years and hadn't biodegraded, but they found a way to add something to it and get it to work. So that's a little more in depth on just 3 of those technologies. Hope you enjoyed that.